Okay, so we've done the xylem-phloem identification at 20 paces thing in laboratory, which you should have found pretty easy. We've talked both in lecture and lab about the way in which xylem and phloem each differentiates from generic vascular cambium cells. Now is the time for us to think a bit more deeply about the mechanisms involved in xylem and phloem transport, which, despite both being transport in a general sense, they are actually very different. One of the crucial differences is related to something we went over in the previous lecture. Xylem tubes are acellular at maturity. That is, when the xylem is actually functioning to transport water and mineral nutrients, the cell part of the xylem is long gone. The cell that split away on the inner side of the vascular cambium was really protoxylem and did little more than just swell to size and occupy the space around which the cell wall formed. And it's the cell wall that was all the plant really wanted. Once the cell wall was complete, the flow of water and minerals through the xylem tube blasted that cellular entity right out of existence. As such, the flow of water through the xylem tubes is actually extracellular, and now is the opportune time for me to specify that the watery compartment occupying extracellular spaces in a plant goes by a special name, the apoplast, which again is the contiguous liquid compartment outside of and surrounding all of the cells in the plant. It's analogous to the interstitial space of the animal cells, the watery medium bathing every cell. Now, the liquid inside the cell's plasma membrane is the cytoplasm, and in plant cells there's often a pretty large central vacuole occupying a fairly large fraction of their volume. The liquid inside the central vacuole is the tonoplast, and its main importance is to keep the cells turgid, which means nicely inflated and pushing up against the cell walls. The turgor pressure exerted by the tonoplast is what makes well-watered plant tissue firm and rigid and crisp, as opposed to the flaccid limp state of wilted plant tissue. In phloem, the main vessel is a tubular structure made of cylindrical cells lined up in a row, kind of like xylem, but in phloem, the cells remain in place. At the ends of adjacent cylindrical cells, their cytoplasms are connected by porous sieve plates, and with many such interconnected cytoplasms, potentially hundreds of individual cellular elements, the result is a long and contiguous fluid compartment entirely surrounded by plasma membrane. This is a symplast, literally together cytoplasm, and in the highly specialized symplastic structure of phloem, each cellular unit, a sieve element, is connected with adjacent sieve elements through colander-like borders with large holes connecting them. These sieve plates, the colander-like boundaries between sieve elements, can be seen in microscopic slides, like the cucurbita stems that we had in the laboratory, which are particularly good places for finding sieve plates. So one major difference between xylem and phloem is that xylem transport of water and minerals is apoplastic, occurring through the tubes that are unconnected with the insides of plant cells, entirely extracellular. In contrast, phloem transport of sugars and the water that sugars are dissolved in occurs through these long interconnected cytoplasms, the symplast, and thus phloem transport is symplastic. Another important difference is the way that xylem transport is usually predictable and always going in the direction from the roots to the leaves. The roots are in the soil, where the water and minerals are, and the leaves are in the air where the water is continually being lost to the world through evapotranspiration. With water coming in continually from below and lost continually from above, it stands to reason that the direction of xylem flow is continually in the upward direction. Phloem, in contrast, is more flexible in the direction in which it flows, though it always transports in a direction away from the sugar's source, where the sugar is loaded into the phloem, and towards the sugar sink, where the sugar is unloaded from the phloem for use in whatever the plant needs the sugar for in that particular location. Phloem transport is from source to sink, where xylem transport is from the roots to the leaves. So that's the preamble, and now we move on to the nuts and bolts of xylem and phloem movement, because it's potentially kind of mysterious. Water doesn't self-elevate, and the xylem is effectively translocating liquid water from bottom to top, and the tops of some trees are more than 100 meters above the ground level. But generally, how do you go about moving fluid through a tube from one place to another? Well, there's a key term that applies here, and it's bulk flow. And this generally applies anytime a fluid moves down a pressure gradient. There doesn't even have to be a tube. If you have high barometric pressure over the ocean and low pressure on land, then wind moves onshore. The fluid in this case is air, which is moving in a mass in the direction from high pressure to low pressure. Offshore winds or downslope winds, like the Santa Anas, that bring great fire risk to us here in Southern California, occur when there's high pressure over the Great Basin Desert, Nevada and Utah, compared with lower pressure off the coast. In order to move a fluid, you have to have a pressure gradient, higher pressure in some part of the system and lower pressure in other parts of the system. Moving air from point A to point B requires that you either generate high pressure at A or low pressure at B, or both. Moving water from root xylem to leaf xylem is no different. It requires a mechanism for generating high pressure in the roots and or for generating low pressure in the leaves. And we have both. You should have this image ingrained into your memory from the lab. We talked about how the endodermis formed a single cell layer wall separating the cortex from the vascular core or stele of the root. The cells of the endodermis are sealed tightly. If they are the bricks in a brick wall that is the endodermis, the mortar in between the bricks is a waxy material, the Casparian strip, that effectively seals off any flow of water in between the cells of the endodermis. In order to get from the outside, the cortex, to the inside, the xylem of the vascular stele, the water must pass through the cells of the endodermis. No water molecule, no nitrate ion, or any other plant nutrient makes it into the vascular system of the plant except through one of the cells of the endodermis. This works out well for the plant for two reasons. First, and fairly obviously I might add, it wouldn't be a great thing for plants to allow stuff from the soil to move into the vascular system of the plant, connected to all parts of the plant, without some kind of vetting. How do you keep soil-borne pathogens, viruses, bacteria, fungi, and toxins of various forms from freely accessing the living cells of the plant? Well, you make sure that the only way into the plant is through a living cell, where the plant has the opportunity to block the passage of bad material from the soil into the xylem. The cells of the endodermis are kind of like the guards at the entry station at a national border, making decisions of who gets let into the country and who must remain on the outside. It's entirely possible for a bad virus that's there in the soil to make it through the root's epidermis, especially in the young root epidermis, which is the most porous and permeable to water. The openings between the cells are large enough for the viruses to gain access to the cortex. Then, once in the cortex, the structure is loose enough such that the virus could make it all the way to the endodermis by passing in the spaces in between the cortical cells. Basically, from the soil to the endodermis, there's nothing keeping a sketchy molecule or virus from getting into the plant. But then comes the endodermis. They will tell the virus, the only way in is through us, and they won't let the virus in. This waxy stuff in between the endodermal cells, the Casparian strip, ensures the security of the root's border wall by making that watertight mortar in between the bricks so that no viruses can sneak through in between the cracks. The other thing that happens with the endodermis is the generation of positive pressure in the root xylem. The plant needs a continual uptake of mineral nutrients like potassium and phosphorus and nitrogen containing compounds. These mineral nutrients come into the roots as ions from the soil. The endodermis and the pericycle, which is the layer just inside of the endodermis, are responsible for the active uptake of these ions, which are taken out of the cortex, moved into the endodermal cells, then into the pericycle cells, and then into the apoplast of the vascular stele, which is basically feeding the xylem. This movement of ions also results in the movement of water. This is because of science, because of osmosis. Think about that time you went to Peru. You went swimming in the Amazon and got a case of diarrhea from the E. coli you inadvertently drank when you were wrestling mano a mano with a freshwater stingray. What happened inside your gut was that the E. coli released an enterotoxin that caused the cells of your lower intestine to secrete large quantities of chloride ion into your gut cavity. The movement of ions from your bloodstream into your gut space brought along with it lots of water because of osmosis. The active transport of ions into the gut caused that fluid compartment, the gut, to have a higher osmotic concentration than the side of the membrane with your cells. Water moves in the direction to equilibrate concentration on both sides of the membrane, which in this case means that water moved into the gut space. More ions are transported into the gut space, and those ions are followed by even more water. Now, your gut space has a finite volume, so the pressure inside your large intestines increases higher and higher until, well, I'll let you figure out the rest. The xylem space inside the pericycle and endodermis is that high-pressure fluid compartment being loaded with ions and osmotic water. The fluid moves via bulk flow from where the pressure is high to where the pressure is lower, and that would mean upwards in a plant that is continually sucking away the water from the xylem in the leaves. The ability of plants to generate enough positive pressure from this mechanism to drive water upwards against gravity is demonstrated by the phenomenon of guttation seen in low-growing plants like this strawberry. The pressure in the xylem is great enough not only to push the water to all parts of the leaves of this little plant, but some of the water actually gets pushed out from the margins of the leaf. I should add that this is something that happens only when the rate of water uptake at the roots is high and the consumption of water by evapotranspiration is low, such as on a cool morning when the soil is wet. Lots of water uptake by the roots, little use of water at the leaves because it's cool, and so there's the extra water getting pushed out by guttation. The positive pressure generated by the roots is limited. You just can't generate that much pressure with water osmotically following ions that are pumped into the xylem. Low-growing plants like the strawberry might see all of their needs met by this positive root pressure, but if the column of water the roots are pushing upwards against is above a certain height and mass, then the weight of the water can overcome the force exerted by the osmotic pump at the roots. You can demonstrate this for yourself by cutting tree saplings at different heights. Cut low to the ground, like at knee level, and the pressure exerted by the roots may be great enough to overcome the weight of the short column of water above it, so the xylem sap continues to push out from the cut sapling. Cut much higher, maybe at eye level, and the weight of the water in the xylem column is greater, and if this downward force is greater than the upward force coming from the roots, no water will come out from the cut, and there will actually be a receding of the xylem, sucked backwards into the cut as the xylem weight wins against the root pressure. Maybe it's cutting at waist level that the cut end of the sapling stump neither pushes water out nor sucks water back. This would basically tell you that the pressure being generated by the roots was equal to the weight of a waist-high column of water in the plant's xylem. If a plant grows taller than waist high, it would not be able to meet its xylem bulk flow requirements with root pressure alone. It would need to complement this with negative pressure or suction exerted from above by the leaves. And this brings us to another slide, or a couple of slides that I had you look at in the lab. The cross sections of leaves, as well as the slide showing stomata on the bottom surface of a leaf in a wet mount. I hope this triggers your memory. The important structures here were the upper and lower epidermis layers, with a waxy cuticle on the outside surfaces to keep the leaf tissue from drying out. Between the epidermis layers, there were a couple of different kinds of mesophyll cells. Mesophyll means in the middle of the leaf, and the layout was different for the corn and the eudicot leaf. This leaf, a eudicot, shows a layer of palisade cells, the palisade parenchyma, packed tightly across the top half of the leaf, and a loosely packed spongy cells, the spongy parenchyma, in the lower half of the leaf. The spaces in between the cells of the leaf are air spaces, and this airspace communicates with the air outside of the leaf via the stomata, singular stoma, meaning mouth in Greek. The last thing to note are the vascular bundles that have single xylem tubes feeding water to the cells of the leaf via capillarity. In the economy of an intensely photosynthetic organism like the leaf, there's obviously going to be the requirement of adequate supplies of the three needed ingredients for photosynthesis. These ingredients are light energy, which comes in abundantly from sunlight, water, and, most crucially, carbon dioxide. You might think, oh, the water is coming in from the xylem, and that's why the plant needs xylem. And you'd be about 0.1% correct. Yes, the water used for photosynthesis does come from the xylem, but the vast majority of water served to the leaves by the xylem does not get photooxidized by an electron-crazed chlorophyll molecule. More on that when you take BIO204. It simply leaves the plant, through the open stomata. Allow me to elaborate. Carbon dioxide, CO2, is currently more abundant in our atmosphere than it has been for the past few thousand years. Your knowledge of this fact is certainly a part of your general awareness of the science-related issues concerning the human condition in our present day. Despite its recent anthropogenic enrichment and continuing upward trend, carbon dioxide is still a relatively rare gas in our atmosphere, comprising between 400 and 450 parts per million. Again, this is high compared with the 280 parts per million from pre-industrial times, but it's still 0.04 to 0.045%, less than 1/20th of 1% of the air around us. This is the same gas that plants need in buckets for the metabolic production of sugar. To put things into perspective, the oxygen gas that you need in buckets to permit your metabolism is there at 21%, over 400 times higher than the CO2 that's needed by plants. Moreover, the process of gas exchange, getting CO2 into cells and O2 out of cells, requires a wet interface between the plant cells and the air compartment containing the CO2. So the outside surfaces of the leaves won't do. They're dry, covered with a cuticle layer that prevents the leaves from drying out on the inside. The gas exchange has to happen inside the leaf between the airspace and the spongy cells that are loosely packed, giving abundant surface area across which gas exchange can happen. But these surfaces have to be wet, and here's where most of the water from the xylem goes. As CO2 is pulled into the mesophyll cells, its concentration in the airspace of the leaf is drawn down. The airspace has to get replenished with CO2 coming in from the outside air via the open stomata. At the same time, the oxygen released by cells into the airspace has to exit the leaf airspace, and this also occurs through the open stomata. Gas exchange between the airspace of the leaf and the outside world is simple diffusion. CO2 is lower inside the leaf after its removal by photosynthesis, while it remains close to the usual 410 parts per million outside the leaf. oxygen O2, which is a waste product of photosynthesis, is at higher concentrations than 21% inside the leaf airspace as a result of gas exchange with the mesophyll cells. Open stomata allow the CO2 to flow in from the outside and the O2 to leave the airspace, and this is necessary in order for photosynthesis to continue. If carbon dioxide drops too low in the airspace, photosynthesis grinds to a halt, and this is because of a weakness in the enzyme that binds CO2. Again, a topic that you'll hear more about in BIO204. Bottom line is that open stomata are key to the continued photosynthesis by the plant. At the same time, however, open stomata allow water vapor to leave the airspace. Again, this is because of diffusion. The air outside is drier compared with the 100% humidity that you have inside the airspace of the leaf. All of the wet surfaces of the mesophyll cells assure that the inside airspace is always saturated with water vapor. As water molecules flock out of the leaf through the open stomata, the loss of water content in the airspace of the leaf is immediately replaced via evaporation off of the wet surfaces of the mesophyll cells. Now, the water lost from the mesophyll cells is restored by water from the xylem, drawn by capillarity from the xylem tubes that reach the leaves. The pull of water away from the leaf xylem by the mesophyll cells to replace the water lost through evaporation creates a negative pressure in the xylem tube. Basically a tiny increment of suction that on its own would seem very weak, but summed over the millions of xylem tubes and hundreds of thousands of leaves in the canopy of a large plant, this is enough to draw water to the top of the xylem. This is the negative pressure on the leaf side, the destination side, that is responsible for the bulk flow of xylem sap, together with the positive pressure generated by roots by the act of transport and the concomitant flow of osmotic water. It's a good idea here to distinguish between two water transfers occurring in the leaf. Evaporation is the phase change of water from liquid to gas as it evaporates off the surface of mesophyll cells and into the airspace inside the leaf. Transpiration is the net diffusion of water from the leaf airspace to the drier air outside the leaf, and this is the part requiring open stomata. Together, they are evapotranspiration, and summed over the entire leaf canopy, this is what drives the suction pump that assists the bulk flow of xylem sap. Now, of the two, root pressure operating from below and negative pressure exerted by evapotranspiration from myriad leaves above, which is the more important? I think there's little debate about this one. The positive pressure pump can account for the lift of water to a little more than a meter above the ground. Plants grow to as high as 115 meters. So if the roots of a sequoia tree push the xylem sap up 1 meter, and the other 114 meters goes upwards thanks to the leaf mechanism, that means they're all even, right? Okay, but thinking further about this, the tallest trees anywhere in the world, that is, living under conditions that are ideal for super tall trees, they all tend to top out between 80 and 100 meters, with a few freaky coastal redwoods going a bit above that. To me, this hints at there being an upper limit on the height to which this mechanism of evapotranspiration can raise water and distribute it to the leaf tissues of the canopy. Try to grow taller than 100 meters, and even this mechanism fails to effectively draw up the water. But when compared with the osmotic pump operating from the roots, where the maximum height is around 1 meter, the leaf pump is definitely responsible for most of the heavy lifting. Now, on to the phloem. Unlike xylem, phloem tubes are, as we said before, cellular, or at least symplastic. They're actually made up of highly specialized cells, the sieve elements, that are connected end to end via perforated sieve plates that make up a contiguous symplast through which the transport of sugars as well as a few other things occurs. The movement of phloem sap is from source to sink. Let's draw this out. So when you look at the phloem in cross-section, you can sometimes spot sieve plates, the perforated ends of the sieve elements. If you slice across the phloem at random, most of the tubes you get will be in the hollow part, but every once in a while you'll pick up the ends of the sieve elements. Okay, so here's the wall of the phloem tube, and here are the perforated ends of the sieve elements. If you happen to get a slice like this on your cross section, that's where you'll see the sieve plates. But most of the phloem you see will have been cut someplace like this, where the phloem will just appear like an empty tube. Okay, the symplast is surrounded by this contiguous plasma membrane and a cell wall, and that's about all there is to the phloem tube, a cell wall and a symplast. There are no normal cellular structures like a nucleus or endoplasmic reticulum because this is primarily a transport vessel. All maintenance functions are handled by companion cells that sit adjacent to the phloem tube. Now, surrounding the phloem, like every cell in the plant, is the apoplast, the extracellular space, and this includes all the water bathing the phloem and imbibing the cell wall. And it's connected to and fed by the xylem. Loading of the phloem occurs at the source end, wherever the plant is producing an excess of sugars to be transported elsewhere in the plant. Now, we usually think about this as the leaves because this is where the sugars are being originally produced via photosynthesis. But the tiny leaves on a growing branch would actually be sugar sinks, receiving more sugars than they're producing, until at least they get close to maturity. Storage organs like tubers or roots may also be sugar sources when their energetic stores are being mobilized to support plant growth elsewhere. So it's not always the leaves. We'll just say source in this diagram. Sugar unloading occurs at the opposite end, the sink end, although there will also be some unloading of sugars at sinks along the entire length of the phloem. If the leaves are the source and the roots are the sink, by the time the phloem actually reaches the roots, there might not be a whole lot of sugar left to unload. The conceptual bit that I like to emphasize here is that the actual movement of sugars from the source end where the sugar concentration is high to the sink end where the concentration is low might actually occur in two distinct ways. First, it's hypothetically possible for the sugars to move entirely by diffusion. That force driving systems toward a uniform distribution of solutes throughout, which would be the lowest energy state, that is, diffusion equilibrium. But because the concentration gradient is maintained by sugar loading continually at the source and unloading at the sink, equilibrium is never reached, and thus this diffusive movement of sugars occurs continually. This purely diffusion-based mechanism is separate from bulk flow because it could occur in a perfectly stagnant, that is, non-flowing tube. Now, the second mechanism here is bulk flow, the physical movement of the fluid within the phloem from the source towards the sink. And this requires a pressure gradient, high pressure at the source end compared with the lower pressure at the sink ends. Now, here's the really neat part. Maybe the neatest part of this whole narrative. The very act of loading the phloem symplast with sugars at the source end results in the osmotic flow of water into the source end as well, because the sugar is an osmotic solute just like any other. Remember the diarrhea? Remember the positive pressure pump at the root end of the xylem? Both were the result of water following osmotic solutes as they moved from one compartment into another. What this means is that the loading of sugars in the phloem will create a pressure head that will push phloem sap away from the source end in the direction of lower pressure. Meanwhile, at the sink end of the phloem, unloading the sugars is removing osmotic solutes, and this will cause water to leave the phloem, reducing the pressure. I hope this strikes you as a totally awesome feature of phloem transport. The pressure gradient that results in bulk flow forms as the result of the loading and unloading of sugars at the two ends of the phloem. As you marvel at this mildly mind-blowing realization, note that this works because the sugars are loaded into and unloaded from a symplast. It's the membrane that isolates the sugars within that causes the water to flow osmotically in the same direction as the sugar. When you think about it in these terms, the fact that phloem translocation is symplastic makes perfect sense. You only need to think about how bulk flow is achieved in the phloem, and you realize that symplastic transport is what makes this mechanism possible. In the next video, we'll revisit the ins and outs of vascular physiology with the help of some very special insect herbivores.